Hello! In today's video, we shall learn in detail about sex determination. By the end of this video, you will have a clear understanding about sex determination in humans, Drosophila, reptiles, Bonellia, and Crepidula. Let us begin with the introduction to sex determination. Sex determination refers to the most fundamental, developmental, biological process by which an organism's sexual trait can be determined. The organism may develop as male, female, or in some species as hermaphrodite. It is a mechanism in which genetic, environmental, or both are responsible for expressing the sexual characteristics of an individual. In humans, sex is determined by specific combination of sex chromosome at the time of fertilization known as genetic sex determination. Certain organisms exhibit temperature-dependent sex determination during critical periods of development, a process known as environmental sex determination. There are different chromosomal systems, namely XY system or XX and XY type, common in mammals. Males are XY and females are XX. ZW system or ZW and ZZ type found in birds. Males are ZZ and females are ZW. And XO system or XX and XO type seen in some insects. Males are XO and females are XX. Now, let us learn about chromosomal sex determination in humans. Primary sex determination is the determination of the gonads. It involves the formation of either an ovary or a testis which occurs within the embryo during organogenesis. Secondary sex determination in humans deals with the development of male and female phenotypes due to the hormones secreted by the testes and ovaries during adolescence. In females, each egg has a single X chromosome, while in males there are two types of sperms, one with X chromosome and the other with Y chromosome. If the X chromosome of the female fuses with the X chromosome from the sperm, the resulting individual is XX, a female which forms ovaries. If the X chromosome of the female fuses with the Y chromosome from the sperm, the resulting individual is XY, a male which forms the testis. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us continue with the topic. The Y chromosome plays a pivotal role in determining the male sex. The gonad first develops through a bipotential stage. In the XY individuals, the primary sex cords continue to proliferate and later form the seminiferous tubules and the germ cells which form the sperms. In the seminiferous tubules are the Sertoli cells which nurture the sperms present in the testis. Anti-Mullerian hormone AMH secreted by the Sertoli cells causes degeneration of Mullerian duct or paramesonephric ducts present in the embryo. The interstitial mesenchyme cells of the testis develop into the Leydig cells which secrete testosterone, causing Wolffian duct to differentiate into epididymis, vas deferens, seminal vesicle, scrotum, and penis. In XX individuals, the internal sex cords degenerate and a new set of sex cords are formed which remain in the cortex and are known as the cortical sex cords. The epithelium of the sex cords becomes the granulosa cells, while the mesenchyme cells become the thecal cells. The granulosa cells and the thecal cells form the follicles that surrounds the germ cells. Each follicle contains a single germ cell. In female, estrogen secreted from the fetal ovaries is responsible for the differentiation of the Mullerian duct into oviduct, uterus, cervix, and upper section of vagina. The Wolffian duct or the mesonephric duct in the embryo degenerates due to lack of testosterone. SRY is the gene that encodes the human testis determining factor. It is located near the tip of the short arm of the Y chromosome. The SRY gene is responsible for initiating the development of male characters. SRY gene is present in normal XY males and rare XX males. It is absent in normal XX females and rare XY females. SOX9 is an autosomal gene which is considered to be involved in sex determination. It is associated with Sertoli cells. In spite of the absence of the SRY gene, if an extra copy of SOX9 is present in XX human, this individual will develop as a male. SOX9 is activated by SRY gene in humans. Transcription factor SF1 is a protein that is also considered to be activated by SRY gene. It is believed to be active in bringing about masculine characters in both the Leydig cells and the Sertoli cells. SF1 is important for testis development and for the regulation of AMH in human. Dihydrotestosterone is a hormone that stimulates the development of male characteristics and controls the formation of the external genitalia. The WNT4 gene is involved in the regulation of Mullerian duct development and may be needed for the ovary determination. It plays an important role in female sex development, fertility, and the maintenance of the female phenotype. Next, let us understand chromosomal sex determination in Drosophila melanogaster. The fruit fly Drosophila, sex is determined by the ratio of X chromosomes to autosomes, as there is no sex hormone. Y chromosome has no role in sex determination but is essential for fertility of male sex, as it contains genes which are capable of forming sperms. If the diploid cell has one X chromosome, the insect is male. If the diploid cell has 2 X chromosomes, the fly is female. In some Drosophila, certain portions of the body are male and other portions are female. They are called gynandromorphs. In these cases, an X chromosome is lost from one embryonic nucleus and therefore results in XO males. High value of X to A ratio in Drosophila activates SXL gene in females with the help of proteins encoded on the X chromosomes. In XY Drosophila, SXL remains inactive due to factors encoded on the autosomes and ensures the male development in the fly. The SXL gene regulates sex determination by controlling the processing of the transformer TRA gene transcript. The transformer TRA gene regulates sexual differentiation in Drosophila females but has no role in males. The female specific TRA gene with the transformer 2 gene helps to produce female phenotype. The DSX gene is transcribed in both sexes in Drosophila. It plays a key role in sex determination and differentiation. If both transformer 2 and female specific TRA proteins are present, a female specific transcript of DSX is formed. If functional transformer TRA is not produced, the transcript encodes a protein that inhibits female traits and a male specific transcript of DSX is formed. If the DSX gene is absent in both XX and XY flies, intersexual genitalia are produced. Next is temperature-dependent sex determination in reptiles. In reptiles like turtles and alligators, temperature influences the development of gonads. Due to high temperature, growth is faster, incubation periods get shortened, juvenile size becomes larger, resulting in larger size adults exhibiting sexual dimorphism. In most turtles, females are larger in size, and it is observed that at high temperature, female turtles develop. In green sea turtles, warmer temperature during incubation period can give rise to female turtles, while cooler temperature can produce male individuals. In red-eared slider turtles, incubation of egg below 28 degrees Celsius will result in the development of male, while above 30 degrees Celsius, egg will develop into a female. In the case of crocodiles, males are larger in size and develop at high temperature. Sex determination in reptiles is hormone dependent. The enzyme aromatase converts testosterone to estrogen. The action of the enzyme can be affected by temperature. The estrogen is responsible for male and female patterns of gonadal differentiation. Next is environmentally controlled sex determining mechanism in Bonellia and Crepidula. In marine form Bonellia, sex determination is influenced by environmental factors, particularly presence or absence of female proboscis. A larva that settles on the sea floor develops into a female. If a larva is attracted to a female proboscis, and settles on it, the larva develops into a male. If an incompletely developed male gets detached from the proboscis of the female, it develops as an intersex. It is evident that the female proboscis produces a hormone-like substance or a chemical signal that attracts the larvae, and the larvae develops masculine characters. In mollusks, Crepidula, sequential hermaphroditism is exhibited. Sex determination is linked to age and size. Small-sized individuals become male and the larger ones become female. Crepidula also exhibit protandry. The young ones remain functional males and produce only sperms. As it matures, a transition to functional female phase occurs. The snails are arranged in stacks, with larger, older female snails at the bottom and the young male settle on top of the female. It is suggested that environment regulates the activity of certain genes, due to which all young ones show male phenotype. During adult stages, it initiates other genes to produce female phenotype. In both Bonellia and Crepidula, sex determination is affected by the position of the individual with regard to other individuals of the same size. So today, we have learnt in detail about sex determination in humans, Drosophila, reptiles, Bonellia, and Crepidula. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.